Okay, everyone, it is 12:31, so I think we should get going. Sit yourself down. It's lovely to see so many people here, and we've also got 28 people on Zoom, so welcome everyone. I'm Georgia Mason. I'm Director of the Campbell Centre for the Study of Animal Welfare. For those of us here in Guelph, I want to begin by recognising that we're on the traditional territories of the Mississaugas of the Credit, who are one of the Anishinaabex people. And this week is a particularly important time to remember this, because as many of you will know, it's Truth and Reconciliation Week here in Canada. And this is a time in which there are various events designed to mark the colonial history of Canada and its impact on Indigenous people. And so I want to highlight a few little things you can do if you want to mark this, particularly focused on Orange Shirt Day, which is on September 30th, and which acknowledges the terrible impact of the residential school system. So here's some things that are happening this week. So Orange Shirt Day is on Saturday the 30th, but on Friday the 29th, the university is organising a walk from the AGO to campus at 9:20 a.m. and then a gathering on Branlin Plaza at 10 a.m. Wear your orange shirts, and you can buy orange shirts from the bookstore, and each one you buy will give a donation to the Woodland Cultural Centre, which is the name of the former local residential school. And then on Saturday itself, there'll be a fire lighting ceremony in Royal City Park down by the river, just off Water Street, and that's at 10:30 a.m. And then if you'd rather do something kind of by yourself, or something that involves a deeper dive into the ghastliness of the residential school system, then the Woodland Cultural Centre organises virtual tours of what was the Mohawk Institute, and they're really phenomenal. I mean, they're harrowing, but they're really worth doing. There's one tonight at 6 o'clock. There's one at 11 a.m. on Saturday. They're a $10 donation, and they're on Zoom, and I'd really recommend them. And obviously the legacy of this system is terrible high rates of infant mortality, and also in the survivors, a legacy of physical and mental health problems, including depression, our topic today. So that's why it's such a serious issue. Okay, so on to today's talk. I'm really proud to introduce Dr. Aileen MacLellan. Aileen is a MITAX Elevate Fellow at the Ottawa Hospital Research Institute, where she's working with Dr. Manoj Lalu and also Mark Avey of the CCAC on how animal care committees can potentially shape experimental design and elements of research during the protocol review process, or how they fail to, maybe. But before she escaped to Ottawa, actually kidnapping a cat on the way, but that's another story. She was in Guelph for a good long while. She did her undergraduate degree here, and then she did her master's and PhD with me, doing phenomenal research on why laboratory mice can sometimes become really strangely inactive, and then developing that question to ask whether laboratory mice and indeed other animals can develop signs of clinical depression like we see in humans. So over to you. One last little thing, if you're here because you're a lab animal vet or tech and you're after CE credits, then give Laura Harper at the back a shout for evidence you were here, and we are also popping her email address online on Zoom. Okay, Aileen, the floor is yours.
All right, so thank you so much, Georgia. I'm just going to put this out of the way. All right, lights are on, we're good. All right, so hi everyone. Thank you so much for being here this afternoon over lunch. As Georgia said, I'm now in Ottawa, but I'm so happy to be back here today and talking about some of the work I did during my PhD in Georgia Mason's lab, investigating the effects of conventional laboratory cages on mice, asking whether these conditions cause mice to be depressed, dysphoric, or maybe simply stressed. But before I get into the nitty-gritty details of how we tested a hypothesis like this, I do need to lay some groundwork. So my plan will first be to go over an introduction, just kind of asking the question, can animals actually develop depression? Then I'll present a framework for identifying animal depression. At the same time, I'm going to go over how we tested this housing-induced depression hypothesis and highlight a few of the methods obstacles we had to tackle along the way. And then, of course, discuss some of the implications of this work. So to start, we've all heard claims about animal depression. This might have been anecdotal claims from your family or friends about their pet dog or cat being depressed, maybe after losing a companion. Or maybe you've been hearing terms like depression or depression-like in biomedical research when animals are being used to model the disorder, typically through manipulations of stress of some sort, and then assessing 1 or 2 signs of depression and often labeling animal responses as depression-like. If you're a welfare researcher or just aware of the literature here, you might have noticed that it's become increasingly common to label animal responses as depression-like. So just as a few of the sort of diverse cases that this pops up in, we're seeing it when we see shelter dogs that are laying inactive and awake, research primates that display hunched postures, working horses in harsh conditions, and riding horses that become withdrawn and display flat neck postures, sows in restrictive conditions that become anhedonic or resist restraint, mice in barren cages that show increased immobility in forced swim tests. And the kind of through line through a lot of these is that we have a habit of classing these states as depression or depression-like when animals are showing low activity or low arousal responses to suboptimal conditions. And I'll flag here that there's certainly variation in the strength and the nature of the claims that are made. But if we're looking specifically at the term depression or depressed, and maybe less so at depression-like or depressive, then it's really important that we distinguish between lay usage of the term. So in lay terms, I would often be referring to a sad or depressed mood that's typically poorly defined or transient. And this is very distinct from when a clinician uses the term to describe someone who's clinically depressed. So clinical or major depression is an affective disorder. It has known risk factors, established signs and biomarkers, and specific diagnostic criteria, importantly. But we also do see bolder claims labeling animals depressed, even in published academic literature. So we see claims like this happen when looking at logging behavior of cetaceans. So for example, Kiska here, who used to be kept at Marineland. We've also seen this term applied to anxious house cats who refuse to eat and are aggressive towards their owners. And also in zoo-kept squirrel monkeys who become reactive and avoid visitors. And all of these cases across the board, these anecdotal reports, these use of the term depression-like, and these maybe more explicit labels, they're plausible because chronic stress is a risk factor for human depression. And all of these animals are experiencing something aversive, but it's simply not enough to conclude that the states these animals are experiencing is depression, and it's certainly not enough to rule out other possible states. So what does it, or what should it mean to label an animal as depressed? Now, this classification is important because if animals are experiencing depression, as in the clinical disorder, this has some fairly serious ethical implications, especially if it's happening in response to conventional conditions. So things like the way that we house or handle our animals that are certainly not intended to induce stress and definitely not intended to induce disorders. But the other reason that this is worth asking is because right now, as I mentioned, we have a tendency to label animal responses as depression or depression-like when they involve inactivity or low arousal. And this may be true. It's possible that these animals are experiencing depression, but it's equally possible with the information we currently have that they're experiencing states like boredom, pain, fear, sickness, things like this that would trigger the exact same response. And that means that right now we could be trying to solve some welfare issues in all of the wrong ways. So if I'm offering resources that are enriching the environment and providing exercise opportunities, this could actually be good if an animal is experiencing depression, but it's not going to be helpful at all if they're actually sick or in pain. So with all of that in mind, if we want to answer this question, we first need to ask what exactly is clinical depression and how do we diagnose it in humans? So clinical depression, as I mentioned, is an affective disorder. It's very complex and heterogeneous, and it has fairly broad impacts. So someone who's depressed will notice changes in behavior, their cognition, and physiology. In humans, we use tools like the Diagnostic Statistical Manual of Mental Disorders, or the DSM. to identify cases of depression. And here, depression is confirmed when an individual shows the presence of 5 co-occurring symptoms out of this list of 9, where one must be low mood or anhedonia. And so this certainly seems straightforward, but the fact is that when we actually look at these combinations, results in some very heterogeneous manifestations of depression. So when we consider all of these criteria, there's actually 227 different symptom combinations that would trigger a diagnosis of depression. And then when we take into account the fact that some of these are bidirectional, so we have psychomotor agitation or retardation, weight loss or gain, insomnia, hypersomnia, this number actually jumps to a staggering 945 unique symptom profiles that we're labeling all of them under the umbrella term of depression. So this means that one case of depression could look drastically different from another. And it also means that things that we don't necessarily expect or intuitively think reflect depression can be. So something like psychomotor agitation, which could manifest as hyperactivity, could actually reflect this disorder. Together, this means that we really can't be trusting our intuition when we're trying to decide whether an animal is depressed or not. And we also can't just be assessing 1 or 2 signs if we want to rigorously test a hypothesis about animal depression. But on top of all of this, in humans, we do often see subclinical cases of depression, sometimes termed dysphoria. And this is where individuals show some signs of depression, but they don't quite meet that 5 out of 9 threshold. And the key thing to remember here is that these individuals still experience suffering, they still have compromised quality of life and often require treatment. So this is another thing that we want to keep in mind, but it does add a level of complexity when investigating animal depression. So given this complexity and heterogeneity, can we actually apply these human diagnostic frameworks to animals? Well, spoiler alert, I'm going to argue yes. But if we want to do this, we first need to distinguish between signs and symptoms. So a symptom is a subjective element that can only be detected through self-report. But a sign, on the other hand, has objective features that can be observed or measured by others. So if I have a sprained ankle, the symptom I experience might be pain, but one of the signs that could be observed would be swelling. Similarly, if I have the flu, I might be experiencing and reporting fatigue and aches, but the sign that a physician could measure would be a fever. And then when it comes to our depression criteria, we also have signs that correspond with the symptoms that we would self-report. Don't worry about trying to sort of cross-reference and take in all of the corresponding signs and symptoms at this point. I'll walk through that slowly later. But what I want to highlight is that while 6 of these symptoms do have corresponding signs, 3 of them don't. So that being feelings of fatigue or loss of energy, feelings of worthlessness or guilt, and then recurrent thoughts of death or suicide. And these we need verbal self-report to assess them because we don't have a reliable corresponding sign right now, so we can't assess them in animals. Which means that we have a total of 6 signs to work with, and we need 5 to detect or confirm depression. So it's doable, but the odds are kind of against us. It's much stricter than the 5 of 9 criteria in humans. But ultimately, even if these odds are stacked against us, if we're looking at signs, we can apply these frameworks. And then if questions arise about how appropriate this might be, just kind of in general, it's important to remember that depression is simply a mood disorder. When we say it, it sounds like something that's just so human and specific to us, and maybe we're being anthropomorphic, but it really is a mood disorder. And if we're willing to accept that animals can experience emotions in longer term moods, which is something we certainly do, we rely on this assumption for several fields of research, including when we use these animals to model disorders, then we also have to be willing to accept that they can experience dysregulation of these systems and develop disorders. So ultimately, this means that yes, human diagnostic frameworks can be applied to animals if we're careful in doing so. But then if this is the case, we then have to ask, has depression already been confirmed in any animal species? And I highlight this because, I mean, as you've already seen, there's lots of claims made about animal depression. And this is from diverse fields of research, but this would include researchers using animals to model the disorder. And here you would have great face and construct validity if you're looking at all of the diagnostic criteria for depression within your model. So there's certainly incentive for researchers to have looked at this. And for welfare scientists, we're also quite motivated to understand the states that these animals are experiencing. And so again, if animals are meeting diagnostic criteria and we have strong evidence that depression is the state they're experiencing, then we might be able to identify more effective treatment and prevention strategies. So with that in mind, we wondered, had anybody done this already? And going in, we knew that there was at least one claim that animals had met diagnostic criteria for depression before. And this came from Ferdowsian and colleagues. And they had assessed DSM criteria in wild and sanctuary living chimpanzees. And the authors concluded that individual chimpanzees could meet criteria for depression, and that some of them did, and that being sanctuary living and having had experienced aversive events was a risk factor for this. These individuals were more likely to be classed as depressed. But this work was met with some fairly intense criticism, particularly because it relied on some subjective observer assessments. So to assess some of these DSM diagnostic criteria, they were using caregiver reports, descriptions of animals having attention or concentration deficits or being easily angered. And some of the measures used were prone to poor inter-rater reliability. So the general approach taken was good, applying diagnostic criteria, fantastic, pragmatic, but the methodology used meant that the evidence here wasn't that persuasive. And even though there's been many excellent reviews relevant to animal depression, the aims of papers that were existing hadn't really been to show evidence of these co-occurring diagnostic signs and animals meeting criteria for depression. So instead, these papers often focus on things like in-depth reviews of how we're modeling depression in animals, maybe evaluating model validity and presenting potential frameworks for testing depression hypotheses, or reviews drawing parallels between captive conditions that we are using and then the ways that we're currently modeling depression in animals. So with this, what was out there still didn't seem like there was evidence of co-occurring signs and symptoms. So we set out to address this gap and look for evidence of co-occurring diagnostic criteria from studies that were using objective markers of these DSM signs. If you're interested in the review that we published, the paper is here and there's a QR code, but of course, feel free to shoot me an email later if you would like to read about it. In this work, we set out to answer the question, does chronic stress and/or poor welfare conditions induce depression? And we selected areas where the term depression-like was already being frequently applied in hopes of kind of capturing this evidence of co-occurrence if it was actually out there. So we looked at rats exposed to chronic unpredictable stress to model the disorder, mice housed in barren conventional cages, pigs kept in restrictive systems, laying hens with keelbone damage, and disbudded dairy calves. And then from existing research, we checked whether these DSM signs had already been confirmed in these cases. So here we found that chronic stress was a risk factor for all of the individual signs in rats. For mice kept in barren cages, the same was true. For pigs in restrictive systems, all of the signs that had been assessed were present, but changes in sleep hadn't been assessed. The same with laying hens, except here changes in sleep had been assessed but not detected. And then for dairy calves, all signs that had been investigated were shown to be present, but changes in sleep and cognitive changes hadn't been assessed. But what this meant was that although from our literature review, we found the presence of 5 or more signs in most of these cases, with the exception of dairy calves, this evidence was pulled from many independent studies. So that crucial co-occurrence of 5 or more signs in an individual still had not been shown. So to the best of our knowledge, no case of animal depression had been confirmed prior to this work. So then the question was, if at present we don't have any confirmed cases, then how exactly can we test this hypothesis? So here we chose to use laboratory mice as our model species. This was because they're well studied, which meant that we had validated indicators of most of the DSM signs we wanted to assess going in, which was great, meant a little bit less background work for testing it. But then the other really valuable thing about this mice as a model species is that conventional laboratory cages, like the shoebox size cage here, are known to restrict highly motivated species typical behaviors. And as a result, they compromise mouse welfare and induce chronic stress. On top of this, these conditions had been shown to increase at least group level risk factors of all of our measurable DSM signs in independent studies, which is promising. But again, I'll emphasize that that crucial co-occurrence of signs in an individual had not yet been demonstrated. And then importantly, well-resourced environments have been shown to protect against these effects, which are at least in female mice. And this is because in male mice, they sometimes become aggressive and resource guard when they're given access to resources-enriched environments, which can potentially negate or reverse intended treatment effects. So for initial testing of this hypothesis, we started with female subjects. And then if we're going to test the hypothesis that conventional cages induce clinical depression in mice, then the predictions here are that conventionally housed mice would show more DSM signs overall, that they would be more likely to show the co-occurrence of 5 or more signs, therefore being classed as depressed, and that they would also show the co-occurrence of 4 or more signs, therefore being classed as dysphoric or showing those subclinical levels of depression. But before it was possible to test for this co-occurrence, we had to address our first methods challenge. And this is that we didn't have a valid test for low mood in mice going in. So we set out to fill this gap by first aiming to validate a judgment bias task for mice. As many of you may be aware, judgment biases are a type of cognitive bias. And these are simply differences in the way that people in positive or negative emotional states interpret ambiguous stimuli. So somebody with anxiety might see a neutral facial expression like the one in the center of this scale and think this person is not happy with me. But if you're in a great mood, having a really good day, you might see that same facial expression and think, yeah, that guy seems like he's in a great mood, or at the very least not interpret it negatively. It's likely that these biases have an adaptive value and are therefore conserved across species, which has led researchers to develop several clever tasks to assess these changes in animals. So in general, judgment bias tasks for animals involve training animals that one cue predicts the presence of a reward, so maybe something like a high frequency tone. Another cue predicts punishment or absence of the reward, so something like a low frequency tone. And then animals can be presented with an intermediate or ambiguous cue, so here maybe an intermediate tone, and their responses to that cue can be assessed. And here, if animals are in a negative state, they'll respond more pessimistically as though they're expecting punishment or the absence of reward. So judgment bias tests like this have been developed for a wide range of species, but none had been validated for mice despite numerous published attempts. So validation of judgment bias tasks requires you to demonstrate that the task is sensitive to deliberate a priori manipulations of affect. So here what we did to manipulate affective state was we used our housing treatments. So mice were randomly assigned to large, well-resourced cages like the one shown above there. And these are highly preferred by mice and known to improve welfare, or to conventional cages like the shoebox cage below, which, as I've discussed, has been shown to induce stress and compromise welfare. Mice from both housing treatments were trained in a go-go task, and here they had to learn that one odor cue predicted a buried high-value food reward, and that another odor cue predicted a buried low-value food reward. And the odors that we used here were either mint or vanilla extracts, and these were counterbalanced across housing treatments in terms of which one was positive and which was negative. Mice would then be presented with an intermediate ambiguous mixture, so a mint vanilla mixture, and then we would assess their latency to dig to see whether they were interpreting that mixture as more like a positive cue as in predicting reward, or more like a negative cue and predicting absence of reward. What we found was that for mice that were assigned with vanilla as their positive odor cue, there was a significant housing effect on latency to dig during our ambiguous trial, which showed that our conventionally housed mice who we knew had poorer welfare were more pessimistic as expected, which was good. That validated our task. And with this task developed, that meant that we now had a proxy for low mood, which meant that we were able to objectively assess a sign that corresponded with each of our 6 measurable DSM symptoms. So we set out to test for co-occurrence, and we did this using 79 female mice that were randomly assigned to conventional or well-resourced cages, like the ones shown here and described from the past experiment. And then we tested them for each DSM sign. For the sake of time, I'm not going to get too far into the details of each test, but I'm more than happy to answer questions later or chat if you have them. But so like I said, to assess low mood, we used our newly validated judgment bias task. To assess anhedonia, this is defined as a loss of interest or motivation for pleasure. And in rodents, this is most commonly assessed through testing for preference for things that are known to be pleasurable. So things like eating tasty treats or mating. So here we used a sucrose preference test, controlling for the normal amount of diet, for the amount of normal diet eaten, so that hunger wouldn't be a confound. In humans, psychomotor agitation and retardation manifest in very diverse ways, but specific forms of inactivity and hyperactivity are common. So here we used home cage behavioral observations, focusing on time spent inactive but awake, like the white mouse in the center there, who's lying still with her eyes open. And we use this as our marker of psychomotor retardation. And then we also looked at hyperactive stereotypic behavior, like the gray mouse who's route tracing in circles around her cage, as our marker of psychomotor agitation. To detect changes in sleep, we also assessed home cage behavior. So here we recorded bouts of sleep when mice were still with their eyes closed for 3 or more seconds. In the video here, there's a gray mouse grooming in the back and a white mouse with her eyes open. She's awake, but there's 2 mice below on a hammock that have their eyes closed and they're sleeping. And then finally, to detect changes in weight, we simply weighed our mice and measured their body length so that we could calculate BMI. And then finally, because inability to think or concentrate often manifests as deficits in working memory in humans, we first used a Barnes maze task to assess spatial working memory, but our sample size for this task was quite small because training was quite time consuming. So we ended up using performance while learning our judgment bias task as an alternate indicator of working memory performance. But then once we had all of these DSM signs assessed in all of our mice, we now ran into our next methods obstacle, which was how do we statistically test the depression hypothesis? Or more specifically, how do we use the continuous data that we have from all of these signs to categorically class DSM signs as present or absent in an individual mouse? And what I mean by this is that when humans report these signs, this is done through self-report during discussions with a physician. Or maybe with checklists or questionnaires. These aren't really simple yes or no categories. So if I tell a clinician that I'm experiencing low mood or I'm having trouble sleeping or concentrating, I'm making a judgment call that what I'm experiencing is abnormal or extreme. But this really does raise the question of how can we do this in animals? How can we categorically class these signs as present or absent? And we have continuous data and we can't rely on self-report. And to illustrate this, if I have a distribution of body weights, the question becomes, at which one of these bars do I say, this is my cutoff, all mice to the right have shown weight gain? And with something like weight, you might say, okay, that seems possible. We have body condition scores, tools like that, which is fair, but it gets infinitely more complex when you think about sleep. How much sleep does a mouse need before you say they're experiencing insomnia? What level of task performance do you need before you say that a mouse categorically has a cognitive deficit? So you can see it's quite challenging. And to handle this issue, we're suggesting that a path forward is to use the well-resourced mouse population to model the general human population. We chose a group of animals because most humans don't experience lifelong restriction the way that conventionally housed animals do. And this is important because using these prevalences allows us to use the prevalence of human depression to help identify thresholds or benchmarks for the presence and absence of signs in our mice. So basically, because we've recommended starting with female mice to test this hypothesis, we would want to be using the prevalence of women, and then we also are looking at the prevalence of depression in adults because our mice were raised to adulthood. And then the other element of this is that because testing for signs of depression is quite labor intensive and so done over the course of a period of months rather than at a single time point, we also wanted to look at prevalence of depression relative to time scale in humans because this impacts it. So what I mean here is that the instantaneous prevalence of depression is 4%. So at any one time, 4% of women would be experiencing depression. But then also if we broaden that, we see higher prevalences. So the annual prevalence for depression is roughly 13%, and the lifetime prevalence for depression in women is roughly 20%. So within their lifetime, 20% of women will have experienced depression. And I'm going to go through this next phase using that 20% lifetime threshold as an example, but just know that we did this for those instantaneous and annual prevalences as well. But our goal was then to set thresholds for signs that resulted in depression classification for 20% of our well-resourced mice, which would match that 20% prevalence in human women. And we did this by assessing the presence or absence of each DSM sign based on the most extreme N percentage of well-resourced mice, and then we could systematically vary this N and check for co-occurrence until we had the prevalence in our well-resourced animals that matched human prevalences. So to give you a better idea of how we did this, my data would look something like this. I'd have a row for each individual mouse, and then a column for each DSM sign with a continuous value going across. From here, if I start with the most extreme 10% of animals, then for each sign, I would go through and mark the most extreme 10% of animals as showing that sign. So the yeses here, the little green squares, this would be individuals who showed the most extreme pessimism in judgment bias tasks or the least sucrose preference and so on. From here, this would allow me to tally up the total number of signs that an individual showed. And that would let me class individuals as depressed if they showed 5 or more signs, dysphoric if they showed 4 or more, and unaffected if they showed less than that. I could then check, once I have these classifications of depressed, dysphoric, and unaffected, whether the prevalence of depression in these well-resourced animals was matching the human prevalence. So if I started with the most extreme 10% of animals for my signs, then I could see, was this resulting in the 20% depression classification for the well-resourced group? If I was falling below that threshold, then I could jump up a notch. So I could say, okay, I will now check the most extreme 15% for each sign and say it's present, check for co-occurrence, and move up from there and so on until I'm matching the human prevalence. Once we had found the thresholds that resulted in depression classification for 20% of our well-resourced mice, I could use these same values to class signs as present or absent in our conventionally housed animals. And then again, check for co-occurrence to class them as depressed. And I recognize that all of this is quite hard to conceptualize when I'm saying it like this. But the main thing that I want you to be able to take away is that our general prediction was that if we're using thresholds that we've developed from the well-resourced population, then we're expecting that all of our individual signs would be present more often in conventionally housed animals, which in turn would mean that we're going to see more classifications of depression in our conventionally housed animals. But if this hypothesis is not supported and conventional housing is not a risk factor for depression, then these thresholds that we're using should yield roughly the same prevalence of individual signs and co-occurring depression classification in both housing treatments. So just a reminder, the predictions of our hypothesis were that conventionally housed animals would show more DSM signs overall, and that they would also be more likely to be classed as depressed and dysphoric. So what did we find? Throughout, I'm just going to present the instantaneous and lifetime values so that you get both ends of the spectrum, but know that the lifetime values showed the same pattern as the annual ones. So here, logistic regressions revealed that for our first prediction, conventionally housed mice did show significantly more DSM signs overall. So here our dark gray or black bars are our conventionally housed animals. They're showing more signs. Great at the instantaneous threshold. And the same was true. We had a significant housing effect at our lifetime thresholds. When looking at depression classification, we had a non-significant housing effect at the instantaneous threshold. So when we have that 4% of well-resourced animals classed as depressed, 6% of conventionally housed animals do. What I want to point out here is that when we're looking at percentages that small or prevalences that small, the number of individuals is also very small. So I'm looking at only 2 of 32 conventionally housed animals and 2 of 47 well-resourced animals. And because we had such small numbers there, that meant that we really didn't have that much statistical power to work with. When we looked at the lifetime prevalence, when we have that higher prevalence, a few more individuals, so a little bit more power, we see a trend for a housing effect, with roughly 31% of conventionally housed animals being classed as depressed when we have 20% of our well-resourced. But then when we turn to dysphoria, we again have a non-significant housing effect at our instantaneous thresholds, again, likely due to low power because we still have just very few individuals. But we do have a significant housing effect at the lifetime threshold. And here, conventionally housed animals are significantly more likely to be classed as dysphoric than well-resourced animals. So taken together, this means that conventional cages increase the risk of depressive signs and can induce dysphoria. And these changes are more prominent when we're using the lifetime or annual prevalence thresholds. And this is important because it contributes to a growing body of literature that shows that conventional cages have severe negative impacts on mouse welfare, and that implementing well-resourced environments should really be prioritized. But the limitations of this work, of course, were low power, as I was highlighting with the number of individuals there. And even when we're looking at those 20% thresholds, those lifetime values, we're still only looking at 9 animals total in our well-resourced group. So that is a challenge to work with. The immeasurable DSM signs, so that 5 out of 9 criteria being much strict in humans, being less strict than the 5 out of 6 that we're working with in animals, really did kind of stack the odds against us. And then the fact that we're unable to truly assess co-occurrence is also a limitation. So unlike if I walk into a doctor's office and say, I'm experiencing this, 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 and this, we simply can't do that with mice. So we had to test these signs over a period of months instead of in a single day to capture that snapshot. So future research should aim to replicate these experiments with a larger cohort of mice to boost power. And maybe down the line, as more automated tests are developed and validated, we could get closer to assessing that true co-occurrence at a single time point. And then one other thing that's worth keeping in mind for the context of this work, and then also looking forward to research of this nature, is that the DSM and the ICD-10, which is the European equivalent, these are imperfect tools for diagnosing depression, even in humans. So there's recognition that these frameworks are relying on subjective reports from patients that are coming in. They involve arguably arbitrary cutoffs for whether a sign is present or absent. They tend to treat disorders as really distinct entities, which sometimes means that we run into problems when there's co-occurrence. And like the other thing that we sort of highlighted at the beginning here is that they can include extremely heterogeneous symptom profiles under this umbrella of a single condition. And this might mean that there's biologically different things going on. The good news is that there's a push within human research to move towards more objective diagnostic approaches, so relying on signs and biomarkers to identify disorders. But this is still sort of in the early phases. If you're interested in this, I encourage you to look into the research domains criteria framework. But for right now, the DSM and ICD-10 are the current gold standard. And as long as this is the case, then they have to be the cornerstone of animal research as well. But looking forward, if and when human diagnostic approaches begin to shift, animal researchers also need to be ready to adapt when they're testing depression hypotheses. But I think this will probably be a welcome change because prioritizing objective signs and biomarkers in humans will just be much easier to translate to animals. So what are the broader implications of this work to close on? Outside of hopefully pushing to improve lab rodent housing, the potential benefits of this work could impact both biomedical researchers and welfare researchers. So if these findings are replicated at least, this potentially presents a more humane approach to modeling depression. So if a cage like this one is inducing a whole suite of DSM signs enough to class an individual as depressed, then this might mean that the approaches that we currently rely on, where we're applying a bunch of environmental stressors over a period of weeks or even months, or we're using intruder paradigms and repeated social defeat or repeated restraint stress, all of these things could possibly be done away with. And instead, we can model depression by simply using the conventional cages that we have as our stressed or depressed treatment group, and using well-resourced environments to house animals as our healthy controls. For welfare researchers, this potentially opens the door to test similar hypotheses across species and contexts. So the sows, hens, and dairy calves described earlier from our review could be an excellent starting point. And again, this opens the door for us to understand the states these animals are experiencing and maybe be more effective when we're trying to improve their welfare. But another potential benefit is that if these standards conditions are causing animals to meet criteria for depression, then it's possible that these individuals could be studied to better understand the disorder. So potentially asking specific questions about underlying mechanisms and things like that, which would mean that we could be using animals that are already in suboptimal conditions, potentially reducing the number of animals that we need to deliberately expose to stressors to model the disorder. And then finally, this work also provides an example framework for researchers that are investigating other affective disorders. And it provides a roadmap for translating human diagnostic criteria to animals. So potential starting points for this could be things like generalized anxiety disorder, post-traumatic stress disorder, and obsessive compulsive disorder. And the overall goal of work like this would just be to be rigorous and improve our construct validity when we're studying these disorders in animals. So with that, I'd like to say thank you to all of you for coming, also to the mice used in these experiments, to our funders, and of course to the Mason Lab, to Georgia Mason for supervising this work, and all of the fantastic lab members that help with these experiments. A lot of careful thought and a lot of hours in the lab went into this, and I'm very grateful. And just before we get into questions, in case anyone's leaving, I will make a plug for you to join the next CCSAW seminar with CCSAW alum Dr. Carly Moody. I don't know if you want to add anything on that now or... Okay. All right. Well then.